Hello guys, today I will be talking about the immune system and its types. I related the fish immune system with humans. I hope you will like the lecture and give your valuable comments in the comments box. Your suggestions will definitely help us to improve in our next presentations. Immunity refers to the ability of an organism to resist or fight off harmful microorganisms, such as bacteria, viruses, and parasites, as well as toxins and other foreign substances. The immune system is a complex network of cells, tissues, and organs that work together to detect and eliminate these harmful invaders, while also recognizing and remembering them to mount a more effective response in the future. Immunity can be acquired naturally, through exposure to infectious agents and the subsequent development of an immune response, or artificially, through the administration of vaccines or immunotherapies. While humans have several major immune organs, including the spleen, thymus, bone marrow, and lymph nodes, fish have a slightly different set of immune organs. Fish have a spleen, which serves a similar function to the human spleen, as well as a kidney, which plays a major role in immune function. Additionally, fish have a unique organ called the thymus equivalent, which helps to produce immune cells called T cells. Fish also have diffuse lymphoid tissue scattered throughout their body, which helps to provide local immune protection. The innate immune system is the first line of defense against invading pathogens and provides immediate, non-specific responses. It includes physical barriers like skin and mucous membranes, as well as cells like macrophages and natural killer cells. The adaptive immune system, on the other hand, is a more specific response that develops over time as the body encounters and responds to specific pathogens. It involves the production of antibodies by B cells and the activation of T cells, which can recognize and target specific antigens. Pathogen recognition receptors are a type of receptor found on the surface of cells of the innate immune system that play a critical role in detecting the presence of pathogens, such as bacteria, viruses, and fungi. PRRs are able to recognize molecular patterns that are commonly found on the surface of pathogens, called pathogen-associated molecular patterns, PAMPs. Examples of PAMPs include lipopolysaccharides found on the surface of gram-negative bacteria, viral nucleic acids, and fungal cell wall components. They respond immediately and no memory or prior exposure is required. In comparison, in adaptive immune system, the receptors generated randomly in B and T cells have a very narrow specificity and can recognize specific epitopes that are derived from polypeptides or proteins, reflecting the individuality of the pathogen. These receptors include B cell receptors, BCRs, and T cell receptors, TCRs, that can recognize and bind to antigens. When a BCR or TCR binds to an antigen, it triggers the activation of B or T cells, respectively, which leads to a slow, 3 to 5 days, but specific immune response against the pathogen. This slow response is due to the need for clones of responding cells to develop. However, the immune system can develop a memory of prior exposure to the pathogen, leading to a more rapid and effective response upon subsequent exposure. The cells of innate immune system include neutrophils, macrophages, natural killer cells, and dendritic cells that can quickly respond to a broad range of pathogens. On the other hand, as discussed earlier, the adaptive immune system involves lymphocytes like T cells, B cells, and antigen-presenting cells, which provide a more specific and slower response to pathogens. Fish and human immunoglobulins differ in several ways. Fish have 3 types of immunoglobulins, including immunoglobulin M, IgD, IgT or IgZ, while humans have 5 main classes, IgG, IgA, IgM, IgD, and IgE. In fish, IgM is the predominant antibody and is produced early in the immune response, while IgT and IgZ are produced later in the response and have specialized roles in mucosal immunity. Additionally, the structure of fish and human immunoglobulins differ. Fish IgM and IgT are pentameric or tetrameric structures, while human IgG is a monomeric structure. Fish IgM and IgT also have a longer hinge region compared to human. As discussed in previous slides, pattern recognition receptors are a type of receptor found on the surface of cells of the innate immune system that can recognize and bind to specific pathogen-associated molecular patterns, PMPs. PMPs are conserved molecular structures found on the surface of various pathogens such as bacteria, viruses, fungi, and parasites. PRRs are germline encoded, non-clonal, expressed on all cells of a given type, and independent of immunologic memory. When PRRs bind to their corresponding PMPs, they initiate a signaling pathway that triggers the production and release of cytokines and chemokines, leading to inflammation and the recruitment of other immune cells to the site of infection. This process is crucial for the innate immune system to rapidly respond to invading pathogens and mount an effective immune response. PRRs can be functionally divided into 3 classes. Soluble bridging secreted PRRs, endocytic PRRs and signaling PRRs. The soluble bridging PRRs include the collectins, ficolins and pentraxins. The endocytic PRRs, which include mannose receptors, scavenger receptors and C-type lectin receptors. Signaling PRRs, which are involved in cell activation in response pathogen. Signaling pathogen recognition receptors, PRRs, are proteins that are expressed on the surface of immune cells and other cell types. These receptors are responsible for detecting the presence of pathogens, such as viruses and bacteria, in the body and triggering an immune response to fight off the infection. There are 2 types of PRRs such as membrane-bound toll-like receptors, TLRs, and cytosolic nod-like receptors, NLRs, and reggae-like receptors, RLRs. You can see in the picture that NLRs and RLRs are mainly located in cytoplasm of host cell. The TLR3 and TLR7 are also located in cytosol, and other TLRs are mainly present on host cell surface. How do innate immunity recognize a pathogen? The answer is simple: by using receptors. Pathogen-associated molecular patterns, PMPs, are molecules associated with microorganisms that are recognized by the immune system as foreign and potentially harmful. PMPs can include components such as lipopolysaccharides, flagellin, and nucleic acids, which are common to many different types of microbes. These are some of the PRRs with their reported ligands or known PMPs. The location of each receptor is also mentioned as reported in humans. At least 20 TLR types, TLR1, 2, 3, 4, 5, M5, S7, 8, 9, 13, 14, 18, 19, 20, 21, 22, 23, 24, 25, and 26 have been found in more than a dozen of fish species. Among all these TLRs, TLR1, TLR2, TLR4, TLR5, and TLR9 are presumed as sensors of bacterial ligands in fish. 6 non-mammalian TLRs were identified in fish. TLR14 shares sequence and structural similarity with TLR1 and 2, and the other 5, TLR19, 20, 21, 22 and 23, form a cluster of novel TLRs. The domain architecture of PRRs can vary depending on the type of receptor, but they typically consist of several domains that are involved in ligand binding, signaling, and/or oligomerization. One of the most well-studied classes of PRRs is the toll-like receptors, TLRs. TLRs are transmembrane proteins with an extracellular domain composed of leucine-rich repeats, LRRs, that are responsible for ligand recognition. The LRRs are followed by a transmembrane domain and an intracellular toll interleukin-1 receptor, TIR, domain that is responsible for downstream signaling. Another class of PRRs are the nucleotide binding oligomerization domain-like receptors, NLRs, which are cytosolic proteins. NLRs contain a central nucleotide binding domain, NBD, and a C-terminal leucine-rich repeat, LRR, domain that is involved in ligand recognition. NLRs also contain a N-terminal effector domain, which can vary depending on the specific NLR and is responsible for downstream signaling. Finally, there are also several other classes of PRRs, such as the reggae-like receptors, RLRs, and the C-type lectin receptors, CLRs, which have their own unique domain architectures. RLRs contain an RNA helicase domain and a C-terminal domain responsible for signaling, while CLRs have an extracellular domain that contains carbohydrate recognition domains, CRDs, responsible for ligand recognition. Overall, the domain architecture of PRRs can vary depending on the type of receptor, but they all contain domains involved in ligand recognition and downstream signaling, allowing for a rapid and effective response to invading pathogens. Here is a nutshell of the TLR pathway. Pathogen recognition. TLRs are located on the surface of immune cells and recognize pathogen-associated molecular patterns, PAMPs, such as lipopolysaccharides, LPS, from gram-negative bacteria or flagellin from motile bacteria. TLR dimerization. Upon ligand binding, TLRs form homo or heterodimers, leading to the recruitment of adapter molecules. Adapter recruitment. The TLR dimer recruits adapter molecules, such as MyD88, which then initiates downstream signaling. Downstream signaling. The MyD88 adapter molecule recruits IRAK1/4, leading to the activation of downstream signaling molecules, including TRAF6 and TUC1. Transcription factor activation. The activation of these signaling molecules leads to the activation of transcription factors, including NF-kappa-B and AP1. Gene expression. Activated NF-kappa-B and AP1 then translocate to the nucleus and induce the expression of pro-inflammatory cytokines and chemokines, such as TNF-alpha and IL-1-beta. Inflammation. These cytokines and chemokines promote inflammation, recruit immune cells to the site of infection, and help to clear the pathogen. Overall, the TLR pathway plays a critical role in the innate immune response by detecting and responding to invading pathogens, leading to the production of pro-inflammatory cytokines and chemokines, and toll-like receptors, TLRs, play a crucial role in linking the innate and adaptive immune systems. When TLRs on innate immune cells, such as dendritic cells, detect a pathogen, they become activated and initiate a series of signaling events that lead to the activation of the adaptive immune response. Specifically, TLR activation leads to the upregulation of co-stimulatory molecules and the secretion of pro-inflammatory cytokines, which together create an environment that promotes the activation and differentiation of naive T cells. These activated T cells can then differentiate into various effector cells, such as T helper cells, cytotoxic T cells, and memory T cells, which are essential for clearing the pathogen and providing long-term immunity. In addition, TLRs can also promote the production of antibodies by activating B cells. TLR activation can induce B cells to produce antibodies specific to the pathogen, which can then contribute to the clearance of the pathogen and provide long-term immunity through the generation of memory B cells. Overall, TLRs play a critical role in linking the innate and adaptive immune systems by initiating the signaling events that promote the activation and differentiation of naive T cells and B cells, leading to the generation of long-term immunity against the invading pathogen. Here is a brief overview of the NLR pathway. Step 1. Pathogen detection. NLRs detect PMPs and DAMPs inside the cell, such as bacterial peptidoglycan or uric acid crystals. Step 2. NLR oligomerization. Upon ligand binding, NLRs oligomerize, forming large protein complexes known as inflammations. Step 3. Activation of caspase 1. The inflammation complex activates caspase 1, an enzyme that cleaves pro-inflammatory cytokines such as pro-IL-1 beta and pro-IL-18 into their active forms. Step 4. Secretion of cytokines. The active forms of IL-1 beta and IL-18 are secreted from the cell, leading to the activation of the immune response and promoting inflammation. In some cases, NLR activation can also lead to a type of programmed cell death called pyroptosis, which helps to eliminate the infected cells. In the next section, I will talk about the cells of immune system. Hematopoiesis is the process of blood cell formation that occurs in the bone marrow in humans, but in fish it occurs in spleen and head kidney. It is essential for maintaining the normal levels of red blood cells, white blood cells, and platelets in the body. The process begins with the differentiation of hematopoietic stem cells, HSCs, into various blood cell lineages. HSCs differentiate into two main lineages, myeloid and lymphoid. Myeloid lineage cells give rise to red blood cells, platelets, and various types of white blood cells such as granulocytes, monocytes, and macrophages. Lymphoid lineage cells give rise to B cells, T cells, and natural killer NK cells. The differentiation process is regulated by various cytokines and growth factors, and abnormalities in hematopoiesis can result in various blood disorders such as anemia, leukemia, and thrombocytopenia. Phagocytosis is a process by which cells engulf and destroy foreign particles, such as bacteria, dead cells, and other debris. This process is an important part of the innate immune system and is carried out by specialized cells called phagocytes, which include macrophages, neutrophils, and dendritic cells. Here is a brief overview of the phagocytosis process. First step is recognition and opsonization. phagocytes recognize and bind to foreign particles through the use of specialized receptors, such as toll-like receptors, TLRs, that recognize pathogen-associated molecular patterns, PAMPs, and opsonins that tag the particles for destruction. Opsonization is a process by which foreign particles, such as bacteria or viruses, are marked for destruction by the immune system. This process involves the coating of the foreign particle with certain molecules called opsonins, which are recognized by immune cells that can then engulf and destroy the particle. Opsonins can be antibodies or other molecules, such as complement proteins, that bind specifically to the foreign particle and make it more recognizable to immune cells. Second step, engulfment. The phagocyte extends its cell membrane around the particle and forms a phagosome, which is a vesicle containing the foreign particle. Third step, fusion with lysosome. The phagosome fuses with lysosomes, which are organelles containing digestive enzymes that break down the foreign particle into smaller fragments. Fourth step, killing and digestion. The lysosomal enzymes then kill and digest the foreign particle, and the resulting fragments are then released into the cytoplasm of the phagocyte. Fifth step, antigen presentation. In some cases, phagocytes can also present the antigen fragments on their cell surface to activate the adaptive immune system. Overall, phagocytosis is a critical part of the innate immune response and helps to prevent the spread of infection and eliminate foreign particles from the body. There are several ways by which phagocytes can kill ingested particles during phagocytosis. Here are some of the main types of killing mechanisms. First type, oxidative killing. This mechanism involves the generation of reactive oxygen species, ROS, such as superoxide anion and hydrogen peroxide, which are highly reactive molecules that can damage and kill the ingested particle. Phagocytes use an enzyme called NADF oxidase to generate ROS. Second type, non-oxidative killing. In this mechanism, phagocytes use various enzymes and antimicrobial peptides to kill the ingested particle. For example, lysosomal enzymes, such as lysozyme, can break down the cell wall of bacteria and fungi, while antimicrobial peptides can disrupt the membrane of the ingested particle. Third type, nutritional deprivation. Phagocytes can also kill the ingested particle by depriving it of nutrients, such as iron or amino acids, that are essential for its growth and survival. Fourth type, apoptosis. In some cases, phagocytes can induce programmed cell death, or apoptosis, in the ingested particle. This mechanism is important for eliminating infected host cells that cannot be repaired and pose a threat to the host. Overall, phagocytes use a combination of these killing mechanisms to efficiently eliminate ingested particles and protect the host from infections. Phagocytes are immune cells responsible for recognizing, engulfing, and destroying foreign particles. There are two types of phagocytes, professional and non-professional. Professional phagocytes include macrophages, neutrophils, and dendritic cells, which are specialized immune cells that have the ability to recognize and destroy foreign particles. Non-professional phagocytes, such as epithelial cells and fibroblasts, are not specialized in phagocytosis but can still participate in engulfing and eliminating foreign particles. Professional phagocytes play a more significant role in phagocytosis than non-professional phagocytes. Inflammation is a natural response of the immune system to infection, injury, or tissue damage. It is characterized by the accumulation of immune cells, such as white blood cells, and the release of inflammatory mediators, such as cytokines and chemokines, at the site of injury or infection. The main purpose of inflammation is to remove harmful stimuli and initiate the process of tissue repair. There are two types of inflammation, acute and chronic. Acute inflammation is a rapid and short-lived response that occurs in response to injury or infection. It is characterized by redness, heat, swelling, pain, and loss of function in the affected area. Acute inflammation is a protective response that helps to isolate and eliminate the source of injury or infection. Chronic inflammation, on the other hand, is a prolonged and persistent response that can last for months or even years. It is typically associated with conditions such as autoimmune diseases, allergies, and chronic infections. Chronic inflammation can lead to tissue damage and dysfunction if left untreated. Inflammation is initiated by the recognition of pathogens or damaged tissue by immune cells such as macrophages, which release cytokines and chemokines that attract other immune cells to the site of inflammation. The process of inflammation involves the activation of several intracellular signaling pathways that lead to the recruitment of immune cells, the release of inflammatory mediators, and the activation of the adaptive immune response. The resolution of inflammation involves the removal of the harmful stimulus and the initiation of tissue repair. The cardinal signs of inflammation are a set of classic symptoms that are commonly associated with the inflammatory response. The four cardinal signs of inflammation are: 1. Redness. Rubber. The affected area becomes red due to increased blood flow and the accumulation of immune cells, such as white blood cells. 2. Heat. Color. The affected area becomes warm due to increased blood flow and metabolic activity. 3. Swelling. Tumor. The affected area becomes swollen due to the accumulation of fluid and immune cells. 4. Pain. Dolor. The affected area becomes painful due to the release of inflammatory mediators and the pressure caused by swelling. In addition to these four cardinal signs, loss of function, functio laesa, is often considered as the fifth cardinal sign of inflammation. This refers to a decrease in the normal function of the affected tissue or organ, which can occur due to the accumulation of immune cells or tissue damage. These cardinal signs of inflammation are a result of the complex interactions between immune cells, blood vessels, and inflammatory mediators, and are an important part of the body's natural response to infection, injury, or tissue damage. Transferrin is a glycoprotein that binds to iron and helps transport it throughout the body. It is an important component of the innate immune system, as iron is essential for the growth and survival of many microorganisms, including bacteria and fungi. By binding to iron, transferrin limits its availability to microorganisms, helping to prevent infection. On the other hand, antiproteases are a group of proteins that inhibit the activity of proteases, which are enzymes that break down proteins. Proteases are produced by many microorganisms as a defense mechanism and are also involved in the process of tissue destruction during inflammation. Antiproteases help to limit the activity of proteases, preventing excessive tissue damage and allowing the body to heal. Here are some enzyme cascades that play critical role in innate immune system. The complement system is a complex series of proteins and enzymes that are part of the innate immune system. It plays a crucial role in defending the body against microbial infections and also helps to remove damaged cells and promote tissue repair. The complement system can be activated through three different pathways: the classical pathway, the alternative pathway, and the lectin pathway. Each pathway involves a series of interactions between complement proteins and cellular surfaces, leading to the activation of a cascade of enzymes that generate a variety of effector molecules, including opsonins, anaphylatoxins, and membrane attack complexes. Now let's talk about lysozyme. It is an enzyme that is found in many bodily secretions, such as tears, saliva, and mucus. It plays an important role in the innate immune system by breaking down bacterial cell walls, making them more susceptible to other defense mechanisms such as phagocytosis. Lysozyme is particularly effective against gram-positive bacteria, which have a relatively thin cell wall compared to gram-negative bacteria. Additionally, lysozyme has been found to have other functions, such as promoting the growth and differentiation of cells in some tissues. The C-reactive protein, CRP, is produced by the liver in response to inflammation in the body. Its levels can be measured in the blood and are often used as a marker of inflammation. CRP is part of the innate immune system and is thought to play a role in recognizing and binding to damaged cells, as well as activating complement pathways and stimulating the production of cytokines. Elevated levels of CRP have been linked to a variety of conditions, including infections, inflammatory diseases, and cardiovascular disease. Cytokines are small proteins produced by various cells in the body that play an essential role in regulating the immune response to infections and other challenges. They can act locally or systemically and have diverse effects on different cells and tissues in the body. Cytokines can stimulate the immune response, promote inflammation, contribute to the development of diseases, and regulate cell growth, differentiation, and survival. They are involved in many physiological processes, including wound healing, tissue repair, and metabolism regulation. The adaptive immune system is a specialized system that recognizes and responds to specific pathogens and foreign substances in the body. It involves the activation and proliferation of B cells and T cells, which can recognize and respond to specific antigens. The immune response leads to the production of antibodies, the killing of infected cells, and the development of memory cells that provide long-term immunity. The adaptive immune system is highly specific and provides targeted protection against a wide range of pathogens and foreign substances. Adaptive immunity displays four characteristic attributes: times antigenic specificity, times diversity, times immunologic memory, times self-non-self recognition. Fish have a specialized adaptive immune system that includes B cells, T cells, and various lymphoid organs, including the thymus, spleen, and kidney. B cells are responsible for producing antibodies in response to specific pathogens or foreign substances, while T cells can directly kill infected cells or coordinate the immune response. The thymus and kidney are primary lymphoid organ where T cells mature, and the spleen is secondary lymphoid organs where B and T cells can encounter and respond to foreign antigens. There are two main types of lymphocytes in the adaptive immune system: B cells and T cells. B cells are responsible for producing antibodies that can recognize and neutralize pathogens and other foreign substances. When a B cell encounters a specific antigen, it can undergo activation and proliferation to produce large quantities of antibodies that can bind to and neutralize the antigen. T cells, on the other hand, can directly kill infected cells or coordinate the immune response. There are several subtypes of T cells, including helper T cells, cytotoxic T cells, and regulatory T cells. Helper T cells can stimulate the activation and proliferation of B cells and other T cells, while cytotoxic T cells can directly kill infected cells. Regulatory T cells play a role in regulating the immune response and preventing autoimmunity. Overall, B cells and T cells work together to provide targeted and specific protection against a wide range of pathogens and foreign substances. There are several subtypes of T cells, including helper T cells and cytotoxic T cells. Helper T cells can recognize and bind to specific antigens presented on the surface of antigen-presenting cells, such as dendritic cells or macrophages. Once activated, helper T cells can stimulate the activation and proliferation of other immune cells, including B cells and cytotoxic T cells. Cytotoxic T cells, also known as killer T cells, are able to directly kill infected or cancerous cells by inducing programmed cell death. They do this by releasing molecules, such as perforin and granzymes, that can damage the target cell's membrane and trigger apoptosis. When a B cell encounters a specific antigen, such as a virus or bacteria, it can undergo activation and proliferation to produce large quantities of antibodies that can bind to and neutralize the antigen. This process, known as humoral immunity, provides targeted and specific protection against a wide range of pathogens and foreign substances. B cells have specialized receptors on their surface called B cell receptors, BCRs, which can recognize specific antigens. When a BCR binds to an antigen, the B cell becomes activated and undergoes a complex series of changes, including proliferation and differentiation into plasma cells. Plasma cells are specialized B cells that produce large amounts of antibodies that can recognize and neutralize the antigen. In addition to producing antibodies, B cells can also act as antigen-presenting cells, which means they can present antigens to other immune cells, such as T cells. This process can stimulate the activation and proliferation of T cells, which can further enhance the immune response. Thus, in conclusion, study of fish immune system can provide us insight of the disease-combating mechanism in fish, which can further be used for producing disease-resistant fishes or fishes with improved immunity. I hope you liked the content of the presentation. Provide your comments about the content you would like to know. This presentation is mainly beneficial for the students who want to know the basics of immune system and its comparison with humans.